A marriage of the two, and Cody's going to talk about torpor and activity in the red-tailed Phascogale.
It's pretty close to 1. Does anyone need a roll? I'm just giving you a chance.
That's your mum, is it?
Shannon? No! So, hi, my name is Cody Noy, and my honours project will be observing torpor expression and also activity patterns in the red-tailed Phascogale. Firstly, I'd just like to explain to you a little bit about what torpor is and why it's so important to the physiology of small endotherms. Torpor can be defined as the significant reduction in metabolic rate and body temperature, and it can be divided into two main groups. Your first ones are your hibernators, which, as you can see from my first graph, which is a function of body temperature versus time, you can have a long hibernating season, which can last for many months. And this is a mountain pygmy possum, and you can see that it spends most of its time with a decreased body temperature at around 5 degrees. And its torpor bouts multiple weeks with short intermittent arousals back to normothermia. So hibernators are able to reduce their metabolic rate to around 5% of their basal metabolic rate, which is the minimum energy production required to remain at normothermia. Similarly, or actually in contrast, daily heterotherms can enter torpor bouts that are less than 24 hours in length. As you can see by the sugar glider here, again, body temperature versus time. The time scale is just 1 month. Each tick is a day. And the sugar glider spends a lot of time warm with daily fluctuations and will occasionally drop their body temperature down to around 15 degrees Celsius. So the differences between them, the metabolic rate, is only able to drop to around 30% of their basal metabolic rate. As torpor involves a significant reduction in the metabolism, it leads to significant energy savings, and it's thought to be induced by factors such as low ambient temperatures and restricted access to food and water. As many of you may know, torpor is quite extensive in the mammals, and specifically marsupials, as you can see by recent phylogenetic studies here. These coloured names here indicate groups in which you have species that exhibit torpor. The blue names here are your hibernators, and your pink are your daily heterotherms. But my study, even though it's broadly across Australia, New Guinea and South America, my study will be focusing on the desiurids down here, which are distributed across Australia and New Guinea. Over half of all desiurids are known to exhibit torpor, and induction into torpor can be spontaneous or induced. Spontaneous torpor means it's a part of the animal's natural daily cycle, whereas induced torpor is a direct response to environmental conditions, such as restricted access to food and water, or like I said before, low ambient temperatures. An interesting point to make though is that spontaneous cycles are not fixed and patterns can change seasonally. A region in which desiurids are particularly diverse is the arid zone, which is interesting because arid zones tend to be notorious for high seasonal fluctuation in ambient temperature, as well as low accessibility of water and food. This is believed to be a result of extensive use of daily torpor, and many species may actually incorporate a process called passive rewarming, which is using exogenous heat like the sun to increase body temperature rather than relying on your own metabolism. As desiurids, such as this Sminthopsis, tend to be nocturnal, they will enter torpor in the early morning and arouse around dusk in the late afternoon. A good example of arid zone desiurids is my red-tailed Phascogale, which is a small arboreal desiurid. As you can see by my geographic range, apart from small isolated populations, these little circles in Victoria, South Australia, and introduced species up here in the Northern Territory, the main trappings of this species occurs in the arid zone here in southwestern Australia. It's an opportunistic predator and it has a diet range of small arthropods to other mammals and sometimes even birds. And the species is sexually dimorphic, with an average male length of around 12 centimetres and a body mass of 50 to 60 grams, whereas females only grow to an average length of around 10 centimetres with a body mass of 35 to 40 grams. Its conservation listing varies depending on who did the observation. The IUCN listing it as near threatened, whereas the EPBC listed it as an endangered species. Previous studies on the red-tailed Phascogale have observed the species entering torpor from an ambient temperature range around 13 to 25 degrees Celsius, with a minimum metabolic rate during torpor of around 30% of its basal metabolic rate. Although long-term data is limited, which is why my study aims to be the first continuous sampling study of the thermal biology of the red-tailed Phascogale. This is important not only in collecting data about a largely undescribed species, but as it's endangered, understanding its physiology is crucial to its conservation. And I'd also like to just broaden the understanding of energy-saving mechanisms of arid zone desiduates. My project is going to have two main experimental procedures. The first, measuring its oxygen consumption and body temperature over an ambient temperature range of around 15 to 25 degrees Celsius. But also, while respirometry is not occurring, I'll be recording daily activity. My experimental procedure has already begun, and it will continue over the next couple of weeks to months to ensure reliable continuous data. I'll be measuring body temperature using these transceivers that were surgically implanted into the animal. They're about 1.5 grams. These transceivers emit a radio pulse, and the frequency between pulse intervals is temperature dependent, with a longer pulse interval indicating a lower body temperature. Body temperature data will be continuously measured during experiments, during respirometry experiments to determine any induced torpor that's occurring, and during intermittent or maintenance days to see if there's any spontaneous torpor that occurs. Metabolic rate, I'll be using open flow respirometry. They'll be placed inside little respirometry chambers and in a temperature controlled environment, which is this fridge, for around 24 hours. During the experimental period, food and water will be withheld to stimulate torpor induction, and they'll be exposed to a natural photo period. I'm measuring activity using a combination of passive infrared sensors and bicycle mileometers. And the passive infrared sensors will be placed on top of the Phascogale's cages and are triggered by movement. The mileometers are placed on top of running wheels, which are included in the cages, and they measure average running distances and speed during the active period. This portion of the study is aimed in observing whether there is any interaction between daily activity and the expression of torpor. I'd just like to finish up by giving you some of the preliminary data that I've collected from my passive infrared sensors. As you can see from the black bars, this is nighttime, activity counts versus time. And you can see that the bulk of the activity is occurring during this dark phase, showing primarily nocturnal activity. I do have induced torpor data, but that's yet to be analysed. Thank you for bearing with my speech. Do you have any questions?
Given that, this is not an unusual thing where zoologists are interfering with animals to try and understand something, but how...
I'm being kind to it. Okay, keep it.
How
It's all ethical.
They told me I could do it, I promise.
How do you know that under your ex situ conditions, your measurements of what you call internal or spontaneous torpor are normal, and how do you control for those sorts of
Well, again, like I said, the data available on torpor in this species is very limited, so I have little to compare it to. But you have to assume that under these conditions, under the
I think I understand the question. Can you try it again? Just
I think though, Jeremy, if he hasn't got, Fritz has got field data on other species, other related species, that should be able to be used as a reference point for the lab data collected on species.
Mind you, yes, up to a point, but then even then, if the data involved in planting things, then how do you know that's not influencing what's happening to
And I have to be careful because the brush-tailed Phascogale is significantly larger.
Anyway, I just put that as a philosophical issue for the
effect.
that
Not in these days.
you would have to do that on a lab.
Yeah.
In plant, you have to measure with different methods. So you've got comparisons between the lab and field. In the field, they do more than the lab. But in the field, it's measured with implant transmitters usually. So that would actually contradict what you're saying, I think, because the transmitters that run in the field. Or you would have to conclude that transmitters actually induce torpor.
No, it's not just the transmitters, also it's just the fact that you're taking them out of them.
No, the thing is, in the field, I was going to torpor more than the lab and publish stuff on that.
And an interesting point is these red-tailed Phascogales are actually captive bred, they're not taken from the wild. So there was no removal from the natural environment in this case.
Then you've got the issue of how much what you're doing, it relates to the wild.
Yes, but it is also an innate part of their physiology. So it's not so much they're learning to go into torpor that they're, in response to ambient temperature, they're actually going into torpor. Maybe they're born.
I was just thinking as well, is there a seasonal effect? Because I know with, for instance, for example, when we do some stuff with them, they might actually, their physiological responses can change between the day and night cycle. So do you, is the day-night cycle you're using consistent all the time? Or
The photoperiod actually, the photoperiod profile changes based on measurements. How long ago was it, Fritz? Of the photoperiod, natural photoperiod in Armadale?
No.
So
you're just using a summer natural photoperiod, or is that?
I mean, it is based on data that was available of the season.
Not changing the actual quality.
Yeah, so yeah, the computer adjusts it based on the date and...
So do you find that, have you found it influences their torpor?
I mean, I've only been doing this...
Well, previously, do you know if the stuff, is there issues with,
There's very strong seasonal change.
Yeah.
Okay, yeah.
As I said before, spontaneous torpor, yeah.
Why are you doing to the local photoperiod? What shouldn't you be doing it to the home range photoperiod?
It's no difference.
Okay, that's all right.
Yeah.
Can you explain that you said the points in Northern Territory that were introductory?
Yeah. They have tried to establish a population of red-tailed Phascogales out near Alice Springs and Uluru. And so they released a number of animals and have been monitoring their growth since they released them.
Was that based on prior knowledge of them being in that?
If I could pull the graph up bigger, there's triangles on my graph that indicate previous fossil records, and I think they have Aboriginal. Yeah. So you have fossil ranges around here where they were known to exist. So yes, please.
Are you going to smoke these guys as well?
I don't plan to.
But that's the next step.
Jeffrey.
What does Paul mean by smoke? I'm intrigued as to what he means.
Like Jay's approach.
Yeah, but the responses to fire.
Oh, okay. Yeah.
On your own.
This is just a basic descriptive thing to see what they do. That's all.
Any other questions? Oh, thanks, Cody. I'd like you to thank all speakers. Again, terrific. An hour and 5 minutes.
Do you mind handing up the mic?
No, it's an hour and 5 minutes. Oh, I think it's